Good morning, everybody. Welcome to our Trainee Research Award presentations. This is the second of 4 of our trainees who are going to tell us about their work today. The 2 we'll be hearing from today are Belinda Wang and Susan Shen. I want to remind everybody that the Trainee Research Awards are we're started to recognize some of the best research that's being done in the Department of Psychiatry and Behavioral Sciences. These are annual awards. If you're doing some fun work, please apply for them in the, generally in the fall. Late fall is when we ask for applications. And then the grand rounds are in the spring. So please, we'd love to hear about all your great work. Thank you for coming and welcome. And I will have Matt come and introduce Belinda.
Good morning, everyone. It's, good to see you. So I am thrilled today. Pretty hot mic here. Thrilled today to be able to introduce Belinda as one of our 4 awardees this year. I have had the great pleasure of officially becoming a co-mentor for Belinda, and so have had a fantastic opportunity to watch what she's been able to do in this, what I think is an extraordinary project that you're going to hear about today. So a little bit about her history. She graduated magna cum laude from Harvard undergrad, and then for unknown reasons decided to stay at Harvard for MD PhD, where then she did her research at Broad, Harvard, MIT, working on cancer and developing a formidable skill set in thinking about how to take a multi-omics approach to tough biological problems. We were thrilled to be able to recruit her here into our research residency. She's had an extraordinary run. I have to say we were thrilled to have her come to our adult residency, and I personally was even equally thrilled, I should say. I don't want to sound too much like a homer, to have her stay on and do child psychiatry training here. She's had a remarkable career as a resident across all of our missions. She's won multiple awards for her science and for her teaching. She's an outstanding clinician. She won the Outstanding Resident Award from the National Institute of Mental Health, the PGY3 Medical Student Teaching Award from our department, the Sorensen Foundation Career Award in Child and Adolescent Psychiatry, and she is a UCSF Weill Clinician Scientist Award winner as well. So she's done extraordinary work while she's been here, and the work that she's going to talk to you about today really is, I think, a tremendous exemplar of bringing together multiple approaches in order, as I said, to attack a very tough but important scientific problem. She has recently led as first author on a manuscript that is now up on bioarchives. Again, you're going to hear about the work today, which is the culmination of a collaboration between the Quantitative Biosciences Institute and the Department of Psychiatry, particularly Jeremy Wilsey, who was her mentor until quite recently, when we sadly lost him to Calico. But this work has really taken a critical step in moving us from a list of genes that are clearly contributing large effect to autism spectrum disorders to the land or the world of proteins, and that, for reasons I think that you're going to see in the talk today, is tremendously valuable, not only in our understanding of biology, but potentially in moving us closer to translation into personalized therapeutics. So congratulations, delighted to play a very small and insignificant role as your recent mentor, and please.
Thank you, Matt. So as Matt alluded to, I've generally been interested in using genetics to understand mechanism of disease, with the big picture goals of improving diagnostic and treatment strategies. So in graduate school, I thought I was going to specialize in pediatric oncology, and so I focused on cancer biology. And I used cell line models to screen how perturbation of each of the 20,000 genes in our genome modified responses to targeted therapy, and found that this information could be used to prioritize subsets of patients that might be responsive or resistant to specific chemotherapy, and also found that we could use this information to identify pathways that could be concurrently targeted to improve treatment response. Then after grad school, to my surprise, I decided to specialize in psychiatry, and I really remained excited about using genetics to understand mechanism of disease, again, with the big picture goal of iteratively improving diagnostic and treatment strategies. So I was thrilled to join the residency at UCSF in 2018, and I was able to join the laboratory of Jeremy Wilsey. And when Jeremy transitioned to Calico, I was very lucky to have Matt accept me into his lab. And today I'm going to share with you the work that our groups have done in which we studied autism risk genes at the protein complex and protein network level to understand how they fit together to drive autism biology. So autism spectrum disorder, which I'm going to label as ASD for the remainder of this presentation, is a neurodevelopmental disorder that's characterized by deficits in social communication and restrictive or repetitive behaviors and interests. It's highly prevalent, and it's among the most heritable of psychiatric disorders. And clinical presentation can be quite heterogeneous. Two individuals with autism can look quite different from one another. We know that behavioral interventions like applied behavior analysis and a variety of support services can be really helpful. But as many of us have experienced, actually getting an autism diagnosis and getting access to these services can be really challenging. And there are currently no pharmacologic treatments that effectively help the core symptoms of autism. And medications are typically used to treat co-occurring disorders like ADHD and anxiety, or are used to mitigate associated symptoms like irritability and agitation. So there's still a lot of promise for a precision medicine approach that links perhaps genetic subgroups of individuals with autism to mechanism-based treatments. So why is it so hard to develop small molecule treatments for psychiatry in general and for autism specifically? I think a big part of the issue is that we don't have the same level of understanding of autism as we do for other diseases like cancer. So a common strategy in drug development is to start with a protein or protein complex of interest that you want to target. Alternatively, you might have an animal model or a cell line model of the disorder that has a phenotype that you're looking to correct. So if we take cancer as an example, this could be, say, the EGFR signaling pathway. This could be uncontrollably dividing cells in a dish. This could be a tumor growing in a mouse. And you can design small molecules or screen existing drugs to bind this protein complex of interest, or which inhibit tumor growth or which kill cells. And we just don't have this level of information for autism. We don't know specific protein complexes to target. We don't really have high throughput cell line or animal models with phenotypes that are clearly related to autism. And then generally en route from DNA to a behavioral phenotype, there are many different levels at which a gene and gene product can work. And for autism, pretty much everything at all of these levels are still being worked out. And today I'm really going to highlight work that we've done in going from genes associated with autism to an understanding of autism mechanism at the protein complex and network level. So as I've alluded to, gene discovery in autism has been enormously successful over the past 15 years. And in particular, sequencing studies in simplex families with unaffected parents and one affected child have been really successful identifying genes that when they have mutations, these tend to confer large amounts of autism risk. And as of today, there's over 200 large effect autism risk genes that have been identified, which gives us a really strong foundation to begin looking for disease mechanism. And I want to emphasize the effect size of these sets of genes. So this graph depicts autism genetic architecture, where on the x-axis is allele frequency, where here are alleles that are pretty uncommon in the population, and here are alleles that are really common in the population. And on the y-axis is the amount of risk for autism that's conferred when you have a damaging variant in these genes. And the group of genes we're talking about is here, where they're not very common in the population, but when they're around, they confer up to 100-fold or more risk for autism. So this represents a really great place to start for experimental studies. Now, we want to go from risk genes to mechanism, and a complication I didn't mention before is that any single risk gene and any mutation will be involved in many different functions at each level. And these functions are going to vary based on how old someone is and what cell type you're looking at. And probably only a subset are going to be relevant to autism. So, for example, let's focus at the protein complex level. There's this blue autism risk gene in blue, and it encodes a blue protein. And let's say that this is involved in 3 protein complexes. How can we tell which, if any of these, are actually relevant to autism? Well, one strategy is to prioritize things that multiple risk genes point to. So for example, if we include information from 3 different autism risk genes, here we might be able to see that all 3 protein products of these 3 genes bind to the same protein complex, but not these other 2. And if we see this, it suggests to us that this particular protein complex might be more relevant to autism. So what we did is we took 100 high-effect autism risk genes and used this as an entry to identify molecular complexes and pathways that are involved in autism. We know that genes and proteins don't work alone. They work in clusters and groups. They form protein complexes. They act in functional pathways. And so we defined protein interactions for 100 autism risk genes with the goal of identifying molecular complexes and modules of interest and trying to figure out how these genes overlap. And specifically what we did is we generated protein interaction networks by expressing tagged versions of high confidence autism genes in HEC293 T cells. So we express these proteins with a tag that allows us to pull this protein out from the molecular soup. And we use mass spectrometry to identify interactor proteins. So this protein of interest friends and who it hangs out with in the cell. And at the end of the day, what we end up with is a protein-protein interaction, or PPI network, where you'll see networks that look like this. And in the center, in a red diamond, it represents the protein encoded by an autism risk gene. And these little dots here in dark gray represent its friends, or the other proteins that they interact with in the cell. So we took these 100 genes and we did this 100 times to generate a protein interaction network for these 100 genes. And we also generated a second network where we introduced variants observed in patients with autism and evaluated how the presence of a patient-derived mutation alters protein interactions, like how do they increase or decrease. And we'll focus first on the network generated from 100 autism risk proteins. So this is a busy network graph. The main takeaway I want to have is, like, it's a big network. We have 100 red diamonds here, which are high confidence autism proteins. And we have a little over 1,000 little blue dots, which are interactor proteins. And they're connected by about 1,800 physical interactions. And of these interactions that we found, actually almost 90% of them are new. So they haven't been defined before in publicly available data sets. So we have this autism interaction network. We have many newly defined interactions, and we had a set of questions that we set out to explore. The first is, of these many new interactions, they were defined in HEC293 T cells, which aren't brain cells. So are these interactors even relevant to the human brain and autism? We looked at expression of autism risk genes and the interacting proteins in human brain expression data. And what we found was that the pattern of expression of these autism risk genes and interactor proteins really mirror each other over the course of brain development. So here on the left to the right, we have periods of development from late first trimester through birth through late adulthood. And we can see that the pattern of autism risk gene expression is pretty high during the prenatal period, dips after birth, and then starts to increase a little bit later in life. And that the pattern of the interactor proteins is really similar. And when we look at individual brain samples, we see that the expression level of high confidence autism genes and interactor genes correlate extremely well across different brain samples. And so from this, we concluded that these interactor proteins and high confidence autism genes they exist in the same space and time in human brain tissue, suggesting that they're brain relevant. We next evaluated whether the interactor proteins that we identified are themselves enriched for autism genetic risk. So we know that they bind to at least one protein that is a high confidence autism gene, but are these interactor proteins themselves, is there genetic evidence for association with autism? And so here we have done a gene set level burden test that evaluates whether interactors themselves are more likely to have a damaging mutation in individuals with autism versus siblings who do not have autism. And we see that the interactor proteins are highly enriched for mutations associated with autistic individuals, whereas other proteins that were present but not picked out as interactor proteins are not. So we really seem to have enriched for a set of genes that are associated with autism. And here we've done a similar analysis where we took the interactor proteins and we asked, are they enriched for different sets of genes that have been newly discovered to be associated with autism? These are gene sets from recent whole exome sequencing studies of autistic individuals. And the last one is a whole exome sequencing study in schizophrenia. And what we see is that the interactor proteins are significantly enriched for autism-associated risk genes that were newly defined, but not for schizophrenia risk genes. And so from this, our conclusion is that interactors are highly enriched for autism genetic risk, but not for schizophrenia genetic risk. And so we believe that these interactions that we identified are highly brain and autism relevant. We've shown that these autism risk genes and interactors are highly co-expressed in human brain tissue, and that the interactors themselves are enriched for autism genetic risk. So with this network that we think is brain and autism relevant, we next wanted to ask, are there molecular complexes of interest within this network? So we found that proteins included by autism risk genes have a lot of mutual friends. So this is another busy network view. And in this depiction, the high confidence autism risk genes are these like blue diamonds in the back, and the interactor proteins are in orange. And the interactor proteins are sized based on the number of autism risk proteins they bind to. So the takeaway I want you guys to have is that there are many large orange dots. So there's several interactors that bind to a lot of autism risk proteins. And one question we had was like, if we just sampled any random set of 100 genes, would we see this? Do proteins just hang out together a lot? And so to evaluate this, we checked the proportion of autism risk genes that share a significant number of mutual friends. So specifically what we did was we pulled pairs of autism risk proteins and looked to see whether they share more mutual friends than you'd expect by chance. And what we found is that about 13 to 14% of autism risk genes share a significant amount of mutual friends, far higher than you'd expect by chance, which would be about 5%. And actually, when we permute from a null model, we observe more like 3 to 4% as what you'd expect if you're pulling just 100 set of random genes. We also compared the connectivity of the autism network compared to two other protein interaction networks that had been generated by Nevin Krogan's group, one for about 40 breast cancer risk genes and one for roughly 80 tyrosine kinases. So these are biologically cohesive sets of genes that we know drive similar biological programs. And what we saw is that these two sets of proteins do have more shared friends than you'd expect by chance, but that the autism risk proteins are far more connected than even these two sets of biologically cohesive genes. So it turns out that about a third of the interactors that we found bind to at least 2 or more high confidence autism proteins. And we observed that several interactors bind to a lot of high confidence autism proteins, like up to 19. suggesting that they might be involved in molecular complexes that are highly relevant to autism. And so we took a look at some of these interactors that bind to 7, 8, or more high confidence autism proteins. And we wound up focusing on one particular complex that involves an interactor protein called DCAF7. Now, DCAF7 binds to 8 autism risk genes. And for practicality reasons, we wound up focusing on this triangle here of DCAF7 and 2 autism risk proteins, DYRK1A and KIA0232. And we wanted to kind of verify that these proteins truly hang out together in cells. And so we expressed these proteins again in HEC293 T cells, and we looked at the mitotic spindle, which is a tubulin-based structure that binds to DNA during cell division and pulls DNA apart. So you can see here in tubulin, these fingers of the spindle binding DNA in the middle, trying to pull them apart. And we see that DCAF7 and DYRK1A also bind to this structure, and KIA0232 seems to form little puncta that also coat this structure. And so from this, we found that all 3 of these proteins do indeed co-localize in cells in the mitotic spindle. They're kind of occupying the same space and time in this cell. We then checked whether perturbation of these 3 proteins lead to similar functional consequences, because this provides us further evidence that these proteins actually act in the same functional pathway. And so we used the Xenopus model system in collaboration with a group of Helen Wilsey with postdoc Michaela Lasser. And Helen had recently shown that knockdown of 10 individual high confidence autism genes lead to altered Xenopus telencephalon size. And she has this powerful model where you can inject Xenopus at the 2-cell stage with Cas9 and a guide RNA. And this allows you to form a hemimutant animal where only half of the tadpole's brain has gene perturbation and the other half is normal, allowing for each animal to be its own paired control. And what Michaela did here was she injected here in these figures, the right half of the brain is perturbed and the left half is unperturbed. And we see that when we use guide RNAs that target a dummy vehicle guide RNA or that target a gene that's not involved in brain development, we don't see any difference between the left and right brain size of these tadpoles. But when you target DYRK1A, DCAF7, or KIA0232, you see a decrease in the brain size on the right side of the animal, which is perturbed. And so here we're showing that knockdown of DYRK1A, DCAF7, and KIA0232 all result in decreased Xenopus telencephalon size, suggesting that these 3 proteins have functional overlap and that they all play a role in modulating brain size. So here, I hope what we've shown is that there are many molecular complexes of interest within this network. We're just looking, we've only looked at the tip of the iceberg. There are many interactors that bind to several autism risk genes. And we've in particular zoomed in on one interactor, DCAF7, and shown that it and 2 autism risk proteins co-localize in cells, and that perturbation of these genes in an animal model result in the same phenotypic outcome. So finally, one other thing that we've been working on is evaluating how patient-derived missense variants affect protein interactions. So specifically, what we have done is we took 54 mutations that were observed in autistic individuals across 30 different high-confidence autism genes, and we generated protein interaction networks using the same method that we used before. And we compared to the original unmutated version of the autism protein to identify interactions that are increased or decreased in the presence of a patient-derived mutation. And what we saw is that most protein interactions don't change in the presence of a mutation, but that there's a subset of interactions that are strengthened or new, and a subset of interactions that are weakened or completely lost. And here is another busy network map. And what I want to highlight is that here in red diamonds, each red diamond represents a high confidence autism protein with a missense variant. The black circles represent interacting proteins, and the color of the lines represent whether in red there are strengthened or new interactions, or in blue that there are weakened or lost interactions. And what I wanted to highlight here in yellow is that there are specific known molecular complexes that have lost interactions with a subset in the presence of a subset of mutations. And there seem to be some shared changes across different high confidence autism proteins. So some examples to highlight. First, we wanted to evaluate whether different variants in the same high confidence autism risk gene lead to convergent protein interaction changes. And what we saw here is that, yes, you do. Here are 3 variants in the protein FOXP1, and we see that all 3 lose interactions with FOXP4. We also want to see whether variants across different high confidence autism risk genes lead to convergent changes. And sticking to the same complex, we saw again that the answer is yes. A mutation in a different autism risk protein, FOXP2, also results in loss of interaction with FOXP4. So it's still early days for the missense protein interaction network, but what we're seeing is that it seems like the patient-derived missense variants do result in convergent interactome changes, and this is true both within individual high confidence autism risk genes and across different high confidence autism risk genes. So to summarize, we've defined a physical interaction network for 100 large effect autism risk genes. We showed that these autism risk genes share interactors, suggesting that they do converge on molecular complexes. We zoomed in on PCAP7 and 2 autism risk genes and found that they co-localize in cells and that they have a shared function regulating brain size. And we're beginning to see that patient-derived variants in autism risk genes lead to convergent molecular interaction changes. And I think the big picture takeaway for me is that it's really cool that autism risk genes converge upon protein complexes. So this is not a case where you have 100 autism risk genes, 100 separate mechanisms, and you'll need to have 100 tailored treatments per individual. And the implications are really that there are shared mechanisms of disease across broader subsets of individuals, which gives us a lot of potential for precision medicine approaches and rational drug design that are applicable to broader subsets of individuals. And some next steps that our group is working on is defining subsets of autism risk genes based on the data. So based on interactor overlap. Perhaps this is a rational approach to stratify patients. We're also working on generating more data. It's both our groups and many others, data from additional cell types and for additional risk genes that have been newly identified. And we think that this approach should and could be applied to other disorders that are emerging with known high effect risk genes, such as schizophrenia and OCD. And the work shown in this presentation involved individuals from many groups, including Jeremy Wilsey, Matt State, Nevin Krogan, Helen Wilsey, and Tom Nowakowski. And I feel very fortunate to have been able to participate in this project as a resident in the adult psychiatry research training program, as well as maintain momentum in the CAP fellowship in kind of a more tailored research track. And I'm very grateful to our department and to my program directors for helping figure out time and funding to do research. And I've been supported by these funding mechanisms, and I can take any questions now.
Thank you, Belinda, for this beautiful talk. So I was curious, like, for example, with the mitotic spindle proteins, I guess presumably some of these proteins are expressed outside of the brain as well. So I was wondering, if there are like deficits in mitoses and other organs outside of the brain, perhaps for these patients.
So yes, these proteins are often expressed widely outside of the brain as well. We haven't looked specifically at like mitotic defects and different tissue types. But what Helen has done, Helen Wilsey has done beautifully recently is she's shown that a lot of autism risk proteins are expressed in non-neuronal tissues, and she's begun to explore overlap in different tissue types and how these might be associated with comorbid features that you see in autistic individuals, things like GI issues, constipation, diarrhea, etc. So I think that you're totally right that a lot of these functions might be non-neuronal as well, and could be also of interest because a lot of these comorbid phenotypes are highly challenging for individuals and families.
Thank you for the talk. This is maybe a question from a novice, but how you mentioned precision medicine in the talk. How far have you gotten, or thoughts about like clinical subtypes of ASD matching to the kind of work you're doing?
I think that it is still somewhat early days. I think that right now, we know that rare variants contribute to probably on the order of like 10 to 20% of individuals with autism. We're moving towards most individuals with autism getting whole exome sequencing. Right now, clinically, that is that doesn't guide primary treatment approaches. But I think that sometime in the next decade, that could change for individuals with rare variants, especially if we move forward in identifying complexes or pathways that are driving disease mechanisms for a subset of individuals. So I think it's, to answer your question, it is still early days. The precision medicine approach is still a bit more of a vision than a reality on the order of a small number of years, but I think that that could change quickly. I hope it will change quickly.